neurodegenerative diseases, assess disease severity, and look at measure cellular response to therapeutics. And so at the time that we devised this imaging modality, we weren't looking to do any of this or even all of this, but I want to tell you how we got here. And it wasn't a very planned path, so just to bear that in mind as we all ideate, we don't know what it's going to end up being, right? And so my lab started out by trying to understand how organelle function impacts cell function by mapping the chemical milieu within organelle lumens. And I want to tell you why, as a chemist, I'm interested in organelles, which are subcompartments of living cells. Because in several diseases, when an organ malfunctions, this malfunction can be traced right down to dysfunctional organelles in the cells comprising that tissue. For example, in heart disease, we know that in endothelial cells, cardiac cells, the Golgi is highly fragmented, right? And now biologists and clinicians have known for decades that of organelle dysfunction in disease. But the reason I feel they have not been able to leverage this fact the way they've leveraged, say, genome sequence information, is that our descriptions of organelles are largely physical and morphological. And I believe as a chemist that until you get chemical measures and molecular measures of normalcy and abnormality in organelles, we will never be able to understand how organelle dysfunction is impacted in pathophysiology and how organelles function in normal physiology. And so that's why my lab developed a platform to quantitatively image chemicals in organelle lumens, right? And this platform really uses just simple short base DNA duplexes that have the function where they can measure ions using fluorescently and quantitatively. And this gives us the ability to interrogate organelle function in what I think are interesting ways. And so now each organelle is actually a reaction vessel, right? It's carrying out different kinds of functions. So each, for example, the ER, okay, the endoplasmic reticulum is making peptide bonds, right? It's joining together amino acids. In the Golgi, we are glycosylating proteins. In the lysosome is built to degrade proteins. In the peroxisome, you have very, very long lipid chains that are being broken down into shorter lipids. And so you'll see that the chemical milieu within an organelle has been optimized over evolutionary timescales to promote the specific biochemistry that occurs within the organelle. And so as I see it, the set of all ion channels and transporters that are present on an organelle gives rise to an emergent property. And that property is to sculpt the luminal chemical environment inside the organelle and promote the unique biochemistry that occurs inside the organelle. And so if you can read out this luminal ionic milieu, then readout of the metabolic activity of that organelle and therefore the metabolic activity of the cell. And so measuring a chemical in an organelle can give you what I call a chemical phenotype. I'm going to call it a chemotype of an organelle. And this chemotype then is a direct readout of the metabolic status of the live cell. And the best analogy I can think of is that if you situate sensors for different temperatures, gases. No, I didn't need that. Oh. I don't want the slide changer. I just needed a pointer. Okay. Oh, there is a pointer. Oh, I just needed to remove it. Thank you. Yeah, so the best analogy, as I was saying, is if you situate different sensors for temperature, pressure, gases in different parts of a car, and you measure these as the car is revving up, you know what normal and abnormal is, right? So the moment one of these levels is completely off, you know exactly what's wrong with the car. We're treating a cell exactly like an engine and putting these sensors in different parts, right, and measuring chemicals. And so today I'm going to talk about chemically phenotyping one organelle called the lysosome. And before I do that, I want to discuss how quantitative DNA-based reporters actually work. So now if you want to measure an ion in an organelle, you need a probe that can perform two functions. The first is if you want to measure your ion, you need a measuring module. And for this, you can use either an ion-sensitive dye or an aptamer for a small molecule or any kind of chemical sensor that's either a turn-on sensor or a turn-off sensor. Because DNA hybridization is 1:1, you have a very precise ratio of sensor chemistry to normalization chemistry. And this allows you to get a ratiometric readout. And a ratiometric readout is always quantitative. So it turns a qualitative detection chemistry into quantitative detection chemistry. The second is if you want to put this measuring module and measure a chemical in an organelle, you need a way by which you can transport this device inside an organelle. And here, we steal from biologists, and we can use the principles of internalization and the complex transport system that exists inside cells. You can co-opt that. And if I summarize all our findings with one concept, let's say I want to target our protein into a target organelle, you identify a shuttle protein on this organelle. Shuttle protein will have two characteristics: high abundance on the target organelle, low abundance on the plasma membrane. Second, there's an active exchange between these two populations. So if you take molecular motifs that bind this shuttle protein, glue them onto your device, then every single guy comes out, grabs a DNA device, takes it inside. After some time, your organelle will basically glow brightly, and it becomes a fancy cuvette from which you can read out the concentration of your ion. And there are a few ways to do this. If your shuttle protein is an endocytic receptor, this is for the aficionados in the audience, simply attach a ligand to the probe. For organelles on the endocytic pathway, because DNA is a ligand for the scavenger receptor, if you're interested in any one of these organelles, simply transfect your scavenger receptor in the cell. If your shuttle protein is not an endocytic receptor and there's no ligand, we've devised a DNA binding protein. It's easy, we can ship it to you, no problem. It's a synthetic protein that binds a very specific sequence of DNA. Simply incorporate that sequence of DNA into your reporter, and it gets trafficked into your organelle. Finally, if you're lucky and people have made an aptamer to your shuttle protein, simply extend the sequence of your DNA to include the sequence of your aptamer, and you can get localized in your organelle. So what is the advantage of transporting a probe into an organelle? So beyond just a simple add an image workflow, the real potential is that you can basically integrate virtually any detection chemistry or any kind of probe chemistry onto this platform, right? Because chemists have made a stunning array of molecules that can sense chemicals and ions and reactive species with high specificities, high affinities, but they're all qualitative. And if you add them to the cell, they'll just paint the cell. So you don't get organelle-specific information. With GFP, you know exactly what organelle you're looking at, very limited chemical information. With our DNA-based technology, we get the best of both worlds. This now becomes quantitative, and you get the stable localization that's afforded by a biologic, and you can simply read out the concentration of your organelle. So I'll just give you a couple of examples of how these reporters work, just to give you an idea. And you could use the same thing to extend it to any one of your detection chemistries or imaging modalities if you're interested. So the first is a pH reporter. This uses a DNA-based sequence that undergoes a conformational change. Stick on two probes that can undergo fret. This fret signal will change with pH and becomes a very nice pH sensor for different organelles. And this was developed by my first PhD student, Shovik. The second is you can take a chloride-sensitive dye. This was made by Workman a long time ago. And Sonali in my lab showed that if you take a ratio, a 1:1, a DNA duplex that's simply displaying an ion-sensitive dye, which is sensitive to chloride, and an ion-insensitive dye, such as Alexa, then that gives you a very nice ratiometric chloride sensor. And here's how you can transport it. You can use this scavenger receptor, as I was telling you. Here's a cell that's been transfected with a scavenger receptor. Simply add your probe, your reporter, to the cell culture milieu. Wait a little time for the cell to internalize it into small endosomes. Wash it off. If you wait a little while, 10 minutes, it's in the early endosome. If you wait 60 minutes, it's in the late endosome. And if you wait a couple of hours, it goes to the lysosome. Remember, I'm going to talk to you about chemotyping of the lysosome. Okay, so we will wait a little while, and I'll show you how we do these experiments. Simply image the cell in the donor channel and the acceptor channel if this is a pH sensor. And if you divide the intensity here by the intensity in the corresponding pixel here, you'll get a number. Simply change that number to some kind of a color depending on its magnitude. And this is essentially now a heat map. So you can basically collect heat maps of lysosomes, pH heat maps, right? So red is a very basic pH, blue is a very acidic pH. Similarly, you can, if you use a chloride sensor, you can get a chloride heat map, right? So you can have, this is yellow or red will be high chloride, and low chloride will look sort of blue. So I'm skipping a lot of experiments, and I'm just going to say that when nobody had known what is the level of chloride inside lysosomes before, and when we finally measured chloride inside lysosomes, we found that it had a highly distinctive milieu. Compared to all other organelles, this organelle had the highest level of acidity that we knew already, and the highest level of chloride. It's even higher than extracellular chloride. And so what we did was we looked at lysosomes that were dysfunctional across a range of diseases, and we found that in all these lysosomes that were dysfunctional in different ways, the chloride values were very low, right? And we found that in terms of pH and chloride, for every proton that's lost in the lysosome, there are 10,000 chloride ions that were lost, right? And so we thought, let's see if we can use this defective ionic milieu to see if we can use it to diagnose diseases. Can we look at disease severity? So that prompted us to make a new kind of sensor that ultimately it turned out to give us 2-ion imaging, which is the technique I'm going to tell you about, which then led to subtyping of neurodegenerative disease and assessing cellular responsivity to a drug. We didn't know that we were going to get there, but I'll tell you how we started. And so far I discussed taking a single sensor, measuring a single ion in a bunch of lysosomes, and then plotting that, right, to give you some kind of average level of ion. So we thought, let's take a single sensor that's capable of measuring 2 different ions in the same lysosome, and it should be able to retain that concentration information on both ions at single lysosome resolution, right? And so for every lysosome, if you're able to plot its value of pH against its value of chloride, could we be able to sort out and chemically resolve lysosomes? That is, do we think all lysosomes have exactly the same milieu? But we didn't know, right? So we just said, okay, let's try and see. Because we know that pH and chloride are independent parameters in the lysosome. We also know from 2D gel electrophoresis and 2D NMR that parameters that migrate, or proteins that migrate closely in one parameter, can be resolved very nicely if you try to separate them on another parameter, right? So can we do that with lysosomes? This is what we were thinking. We had no idea that we would be able to even see another population of lysosomes, right? But anyway, we made this sensor. We basically took the pH sensor and integrated a chloride-sensitive dye on this. So here, this is a FRET sensor. You're going to measure D. You're going to measure A, acceptor. So you'll excite the donor, measure donor fluorescence and acceptor fluorescence. Here you'll measure you'll directly excite the chloride-sensitive dye, measure green, directly excite the red dye, and you'll measure red. So this ratio is going to give you chloride. This ratio is going to give you pH. And we found that neither module interfered with the sensing of the other module, right? So it was you could get a dual functionality on a single scaffold. So your data is going to look like this. Here, we've labeled cells, lysosomes of cells, and what we did was we fixed the pH and the chloride to specific values. This is just a test to see if the system's going to perform correctly or not. We fixed the pH to pH 4 and the chloride to pH to I think 50 millimolar, and then we should expect all of them to be very homogeneous distribution. So this is actually about 600 lysosomes together, but many of them are sitting one on top of the other, right? So we just converted this into a heat plot, where red shows a large number of lysosomes, and blue is a scattering of lysosomes, right? It's not very, so this is a density plot, and I'm going to call this a 2IM profile, because the method, we're calling it 2-ion measurement, right, 2IM. So this is a 2IM profile, and the 2IM profile is a very nice spherical thing. That's what we expect it to be. Things are working as they're supposed to be working. Okay, so now let's take a skin biopsy from a healthy individual and just ask what's the natural distribution, right, by a 2IM profile. And we see that there are at least two populations. One is a very high chloride population, and the other is a lower chloride population. Okay, let's take a couple of other healthy individuals, fibroblasts from 2 or 3 different individuals, and they all have this high population, high chloride population. So we then said, okay, let's take a look at skin biopsies of Niemann-Pick skin fibroblasts that are derived from children with Niemann-Pick disease. So for 50 years, Niemann-Pick disease had been thought to be a single disease. Then with the advent of gene sequencing and things like that, we found out only in the 90s and 80s that it was actually 3 different or 4 different diseases, right? There's A type, B type, C type, and D type. I've just shown you A, B, and C here. A and B are essentially the same disease. It's the same protein that's mutated. It's a protein called acid sphingomyelinase. And A-type people it's very severe. The children basically, it's manifested in the brain, and the children die by the time they are 3 or 4. B, you have a little better, the mutations are milder on the protein. It's manifested in the visceral organs, and the children can live a little longer, maybe up to teenage or so. And Niemann-Pick C disease, it's a totally different protein that's mutated, right? It's got to do with cholesterol metabolism. So what we found was skin biopsies of all of these kids showed that suddenly this entire profile collapses into a monodispersed population, and we lose this high chloride population, okay? Okay, so then we said, okay, let's try and see. Niemann-Pick A and B people have a therapeutic. It's the enzyme called acid sphingomyelinase. This is called enzyme replacement therapy. So literally what they do in a dish to see if therapy is working or not is that they'll take this recombinant protein, they'll add it to the cell endocytoses this protein, sends it to the lysosome where it does its job, right? So this is the therapeutic. And if you add acid sphingomyelinase to either of these Niemann-Pick patient cells, you recover this high chloride population. Niemann-Pick C people have no therapeutic. The only one that seems to be somewhat doing something is something called beta-cyclodextrin, right? But they're still trying to push this through clinical trials, even though they know this is a bad drug. There's enough amount of literature which says this is really bad, but they have nothing else. So you can see here that when we treated each of these 3 patients' cells with the drug, this guy's cells responded somewhat, this guy is a non-responder, and this guy's cells responded somewhat, somewhere on the borderline. If you add this therapeutic to Niemann-Pick A, B cells, right, people who've got Niemann-Pick A or B disease, and this is actually not the therapeutic for these guys, right? You see that there's no response. So with this, you can tell whose cells are going to respond to a therapeutic and whose cells are not going to respond. And so the NIH just got in touch with us after they read our paper. They read it only last month, even though it came out in Jan. They're just starting a clinical trial for Niemann-Pick C disease, and they wanted to use our test to see if we could just follow all the 50 people on this drug and see whether our test can give them some kind of positive predictive values or negative predictive values. And so I'm quite excited by what we're going to see. This is my last slide. So in summary, a simple DNA duplex that carries fluorescent detection chemistry on one strand and a normalizer dye on the other strand can expand qualitative detection chemistries to quantitative regimes. By sticking it on a DNA duplex, which has signatures and molecular GPS, you can achieve organelle-specific information. And so I see this simple DNA duplex as a powerful lens through which one can visualize new biology. And the 2 areas I see this technology having an impact in discovery biology, and I didn't talk about this application here, personalized medicine, which I talked about at length, And finally, we expanded this technology to directly map enzymatic cleavage in organelles. Happy to discuss that later. And all of this was made possible only because of the quantitative nature of the technology, where we can provide numbers and concentrations. I'm going to close with a quote of an Irish-Scottish scientist, physicist, and engineer as well, who never minced his words. And he said, when you can measure what you're speaking about and express it in numbers, you know something about it. When you cannot express it in numbers, your knowledge is of a meager and unsatisfactory kind. It may be the beginning of knowledge, but you have scarcely, in your thoughts, advanced to the stage of science. I'm backed up with a fabulous team, and happy to take any questions.
Well, thank you, Yamuna. So the Scottish-Irish physicist was hoisted by his own petard because in 1895 he said there would never be manned flights. So
yes.
You should have used some numbers and he
Yes.
might have got it right.
Yes, he said a lot of right things and a lot of wrong things. And that's why he said he never minced his words. Right?
Yeah, but don't we all do that,
Yeah.
to be honest? Right.
Yeah.
Sizes of the lysosomes?
Lysosomes are typically about, so they will vary from cell to cell. The largest lysosomes that I've seen are in coelomocytes inside C. elegans. Those can go up to 5 microns. Those can go up to about 5 microns. Smaller ones can be about 200 to 400 nanometers.
How do you know that you're looking at a single lysosome with fluorescent microscopy?
Well, because we do 3D imaging, right? So you can see, but then you can always say that there is one very, very small lysosome sitting on another larger lysosome, and you can come up with anything. So to some extent, if knowing the average size of a lysosome and knowing the pH in the compartment, you can tell whether it's a single lysosome or not. Just ask, it's fine. Yeah.
So the data for the patient's skin biopsy,
Yes.
so these 2D plots,
Yes.
the question is that microscopy data?
Everything is microscopy.
Okay, that's
So,
microscopy data. And then
yes.
the axes,
Yes.
is that the constant?
Yes. So the axis is the raw data. Okay. So this is basically raw data. What we're doing is we're plotting D by A, which is a measure of pH out here, R by G, which is a direct measure of chloride out here. Right?
You can calculate the precise pH.
For sure, you can. Yes, you can calculate the precise. But the thing is, we wanted to keep this so simple to basically idiot-proof the technology, so that anywhere across the world, whoever it is, you just do this one thing, and you'll be for that microscope. We've done it on different microscopes, different skin biopsies. It's always there.